Okay, so ladies and gentlemen, you are welcome. So in today's video, we are going to look at the enzymology and the protein purification. So what is enzymology? Enzymology is the study of enzymes, which are biological catalysts that speed up chemical reactions. So generally in living organisms, there are thousands of the reactions that are taking place, and some of these reactions are taking uh, milliseconds to occur. And of course, in the absence of these enzymes, some of the reactions may take even days, months. So therefore, the main importance of the enzymes in our body is to speed up all those biological processes for proper functions of the body and sustaining life processes. And these enzymes are crucial for various biochemical processes, including metabolism and DNA replication. When we said metabolism, of course, is the spectrum. So metabolism is a sum total of all reactions that are taking place in the body, which include anabolism, metabolism, and amphibolism. And of course, DNA replication is a process in which the DNA converts itself to another molecule of DNA, and it involves so many enzymes and proteins. So the enzymes are actually very important in actually converting one molecule of DNA into another DNA. And of course, the DNA replication is the origin of the life processes because cells only divide when there is a DNA replication. So understanding enzymes helps in the field like medicine, biotechnology, and environmental sciences. And more importantly, how these enzymes catalyze the reaction is, of course, generally before reaction takes place, there is what you call an activation energy. And this activation energy, this is it here. So before the reaction takes place, of course, the reactant must be actually jump this activation energy before the reaction takes place. That is what usually happens. So this is actually in this A, it is an example of uncatalyzed reaction that is in the absence of enzymes. So you see that the activation energy is a huge hill. It is a huge. Look at it here. It is very huge. So meaning that before the reactant molecules converted into product, they must jump this activation energy. But of course, look at it in the presence of enzymes. That is when the enzymes catalyze the reaction. Look at how the activation energy decrease. So therefore, the reactant can just easily converted by just a small amount of energy. They will jump to produce the product. So therefore, enzymes are very important in reducing the activation energy. So that is how they increase the speed of the reaction. So they speed the rate of the reaction by decreasing the activation energy. And understanding the concept of enzymology is very important in medicine because understanding the concept of enzymology, it helps you in drug design, it helps you in drug development, and it also helps us in disease diagnosis as well as the disease treatment. Of course, biotechnology is a technology that trying to produce a product using a living organisms or a part of living organisms like the enzymes itself. It's a part of living organisms like a cell, like a tissue. So to use a tissue or a particular organ to produce a product is what we call a biotechnology. So therefore, enzymes have actually helped in producing a lot of biotechnological products like recombinant insulin, recombinant adrenaline, and so many others. Of course, in the environmental sciences, you can use enzymes to remediate the soil because there are so many contaminated soil. So you can use enzyme to remove contaminants so that to clean the environment. So, ladies and gentlemen, now let's look at the structure of the enzymes. So generally, enzymes are primarily proteins that have a complex three-dimensional structure. So this is actually a typical examples of a complex three-dimensional structure of an enzymes. And in the enzymes, it has what we call an active site. So an active site is a region where it's a part of the enzymes where the substrate binds. Like for example, this is the active site and this is the substrate. So the substrate here will now bind to the active site, and therefore we'll have an enzymes substrate complex. So when we said enzyme substrate complex, it means that the enzymes and the substrate binds. And that is where we have the enzyme substrate complex. So therefore, the enzymes will now convert the substrate into product, and of course, the product will be released, will leave to the active site. So this is what we have. So these are the substrate, these are the product. That is why if you look at them here, the substrate is just a single molecule, while the products are two molecules. In fact, their color are different. So this is to tell you that the enzymes have really converted the substrate into product. So the structure of the enzymes, their structure is vital for their function, as the active site must specifically bind to substrate. So this is actually the structure of the enzyme. So in a situation where the enzymes loses its three-dimensional structure, when it loses its three-dimensional structure, it is complex three-dimensional structure, cause the integrity of the enzymes will be distorted. Therefore, the activity of the enzymes will also be distorted. So therefore, the three-dimensional structure can be distorted by maybe increasing the temperature of the environment where the enzymes is, increasing the pH, increasing the salt concentration. So these are some of the factors that can affect the three-dimensional structure of the protein. So once you have actually a factor that distort the three-dimensional structure of protein, that process we call enzymes denaturation. So once enzymes loses its three-dimensional structure, the process is called enzymes denaturation. So once it's denatured, then the enzyme's activity will not be sustained. And of course, some enzymes contain non-protein components. So generally enzymes, from our basic knowledge, 99% of the enzymes are protein. And in fact, one important thing that we should understand that we have some enzymes that have a non-protein component. And this non-protein component we have as a cofactors or coenzymes. The cofactors can be a metal. So those enzymes that need coenzymes or cofactors to become active enzymes, they are very important to know them. So if there is no these cofactors and there is no these coenzymes, they are called apoenzymes. If they doesn't actually attach with the cofactors or coenzyme, they are called apoenzymes. But once those that actually need the cofactors or coenzymes combined with them, that is when you have coenzymes plus the apoenzymes, then you have holoenzymes. So the holoenzymes is the active enzymes, while the apoenzyme is an inactive enzyme. So if it requires a cofactors or a coenzyme, so when the apoenzymes bind with the cofactors or coenzymes, they'll become the active enzymes. So what are the mechanisms of enzymes action? So as I said previously on the previous slide, enzymes actually lower the activation energy required for the reaction to occur. So the lower the activation energy, as I mentioned, you see this is the reactant here, and this is the product. And of course, here is where the activation energy is lowered. So you see lower activation energy is using a catalyst. So when you use a catalyst, the activation energy will be lower. So the enzyme substrate complex is a temporary interaction. Yes, it's a temporary interaction because after some time it will dissociate, which leads to the product formation. So this process often follows the lock and key or the induced fit models of enzymes action. What is a lock and key? Like the enzymes and the substrate is like a padlock. The enzyme is your padlock, and then the substrate is the key. So you see that the substrate is always perfectly fits with the substrate, sorry, with the enzymes. So it means that when we set lock and key, it means that the enzymes are very specific, only bind with their substrates. And in the case of induced fit models, it's a model that's trying to tell us that when a substrate approach an enzymes, then the enzymes will now change its shape so that the active side of the enzymes will be perfectly changed and aligned with the structure of the substrate. That is the induced fit models. It means that the enzymes will change its shape perfectly fit with the shape of the substrate, and then the substrate will bind for the enzymes catalysis to take place. So that is why we should understand that in enzymes catalysis, it is very specific. It is like a padlock, lock and key. If a particular substrate is not meant for a particular enzymes, then there is no binding and therefore no reaction. So enzymes kinetics. So when we said enzymes kinetics is a study, or enzymes kinetics is always trying to study the rates of enzymes catalyzed reaction and how they change with varying conditions. So enzymes kinetics is a study, or is a studies that trying to show how fast an enzymes will catalyze a particular substrate to product. Like for example, in 1 minute, how much of the enzymes will be converted into product? So enzymes kinetics is trying to tell us the rates, the time in which the substrate can be converted into product. So the Mark-Lewis-Menten equation is a fundamental model that used to describe the kinetics of many enzymes. So that is why the most important thing in enzymes kinetics is to understand the Mark-Lewis-Menten. And this is actually V. This is V. The V here is a velocity, and it is actually trying to show the Mark-Lewis-Menten. So this is a Mark-Lewis-Menten equation. Vm times substrate concentration divided by Km plus the substrate concentration. The Vm here stands for the Vmax, that is maximum velocity. The S here in the concentration bracket is called substrate concentration. Km is a kinetic parameter. So when an enzymes bind with the substrate, the catalysis increases with time until when it reaches a stage where all the active site of the enzymes is occupied with the substrate. Once you reach that stage, then it will now give you like a slope, a plateau slope, where, like for example, let's say this is your uh, coordinate. So let's say you have an enzyme. So what happens when you have an enzyme and the substrate? The speed and the velocity will be increasing as you are increasing the enzymes with the substrate. So it will reach to a level where the active side of the enzymes is all occupied with the substrate, and that is where it will give us this plateau. What this plateau means, it means that the enzymes reach its maximum velocity. That's why we are using Vmax. And then the Km here, the importance of Km, it shows the affinity of the enzymes toward the substrate. And the lower the Km value, the higher the binding affinity of the enzyme. So assuming now you have two enzymes 1 and 2. So the one with the lower Km value, it means that it has more activity than the one with the higher uh, Km value. So this is Mark-Lewis-Menten, the one that I just highlighted in red. Then there are factors that can also affect the enzyme's kinetics. Increasing the substrate concentration can also increase the speed of the reaction. Also, increasing the temperature can also increase the rate of the reaction. Also, increasing the pH. But generally, most of these enzymes, they are specific in terms of their temperature and pH. Most of the enzymes, they work maximally at our body temperature, which is 37 degrees centigrade. So what if the temperature is below that 37? It means that the enzymes will become inactive. And if it is above that, then the enzymes will be denatured. So once the enzyme is denatured, it means that it loses its enzymatic activity. And of course, in the case of pH, most enzymes work at a uh, most enzymes work at neutral pH, and some work in acidic, while some work in alkaline. So if the pH of an enzyme is distorted, it can also lead to the denaturation of the enzymes. So what are the enzymes regulation? That is how you can control the enzymes. You can decide to increase the enzyme's activity, you can decide to decrease the enzyme activity, and you can even decide to off it or on it. So enzymes are regulated through various mechanisms, including allosteric regulation and feedback inhibition. And this is actually some of them here. We have allosteric regulation, we have reversible covalent modification, we have proteolytic cleavage, we have feedback regulation, we have regulation by isoenzymes. So let me just use this medium to actually describe an isoenzyme. So when we said iso means this term. So isoenzymes, it means that there are enzymes that may be of different origin, that is coming from different organs or different cells, but they are all different organelles of the cells, but they are catalyzing the same types of reaction. Like for example, we have glucokinase and hexokinase. Glucokinase and hexokinase are enzymes that catalyze same types of reaction. So this is actually some of the process that you can actually exploit to regulate the activity of an enzymes. To regulate the activity of an enzymes. Like for example, when we said allosteric, so generally, what happens in the case of allosteric regulation, most of the enzymes have another site apart from an active site, and the site is called allosteric site. So allosteric site is a site where a molecules combine to an enzymes, and once they bind, it depends on the types of the molecules. It can be activator, that is the molecule that can increase the activity of the enzymes, or inhibitor, that is the molecules that can decrease the activity of the enzymes. So allosteric regulation is a regulation in which an enzymes have allosteric sites where activator or inhibitor of the enzymes bind to. And then the next thing is reversible covalent modification. Some of the enzymes on their own, they are inactive. So they need a particular group to attach to them or a particular group to remove on them. Like for example, some enzymes are inactive until when they undergo methylation, that is addition of methyl group or addition of phosphate group. So that is a covalent modification. And we have proteolytic cleavage. So in the case of proteolytic cleavage, remember from the basic knowledge of protein digestion, we have an enzyme called pepsin. So enzymes, pepsin is mostly produced as an inactive form called pepsinogen. So it undergoes proteolytic cleavage. That is, it is cleaved by an enzyme by hydrochloric acid and make them active. And that is what we call proteolytic cleavage. We have feedback regulation where the end product of a particular reaction will now inhibit the first enzymes, because most of the metabolic reactions, most of the reactions that are taking place in our body, they are in sequence, they are in chain. You can have 10 reactions in one particular pathway. So the end product of one pathway, like let's say glycolysis, there are 10 reactions. So if you take the end products, which is pyruvate, can now come back and inhibit the first enzymes, and that is what we call feedback regulation. And then regulation by isoenzymes. So you can decide to regulate a pathway by using an isoenzymes of the same reactor, of a particular pathway. So allosteric enzymes have a site where a molecules combine causing changes in activity. And the change in activity, as I said, depend on the molecules can be an activator or inhibitor. And of course, in the case of feedback, the common regulatory mechanisms where the end product of a pathway inhibits the upstream enzymes. So what are the types of enzymes or classification of enzymes? Enzymes can be classified into six major categories. And these categories, we have them here. We started with oxidoreductase, transferase, hydrolases, lyases, isomerase, and ligases. So these are the six classes of enzymes, and each of these enzymes have their specific types of reaction that they catalyze. Like oxidoreductase is catalyzed oxidation reduction reaction, that is redox reaction, and the transferase enzymes it catalyze group transfer where maybe a phosphate or methyl group is transferred from one compound to another. Hydrolysis, it means it actually catalyze hydrolysis reaction. That is, it can be transfer of uh, functional group to water. Lyasis, it means the addition or removal of a group to form uh, double bonds. It might not be necessary to be double bond. It can be a cleavage, that is to break down something into two or more. Isomerase, there are enzymes that catalyze isomerization reaction, and the ligases, there are enzymes that actually describe or that are used in joining two molecules or more together. So each enzyme type plays a specific role in metabolic pathways and cellular processes. So then in briefly, we have what we call enzyme purification. Assuming now you have these enzymes in a particular organelle of the cells and you want to isolate them. So protein and enzyme purification are the same thing because enzymes are protein. So you can actually isolate enzymes or a protein is the same thing. So protein purification and enzyme purification is the same. So assuming now you have an enzyme in a particular organs, let's say in the liver, so how are you going to isolate the enzyme? So there are a series of protein purification steps. So let's say the enzymes is in the mitochondria in human, for example, let's say in a mitochondria in a human. Although it's depend on the enzymes that you want to isolate or protein that you want to isolate, you need to check from the literature, the sources, where do you have that enzyme? Is it present in the human organ? Is it present in microorganisms? Is it present in plant? So generally, microorganisms are the organism that is actually the best to isolate enzymes from them or protein. But if you don't have it in microorganisms, of course, with the advancement in biotechnology, you can undergo um, recombinant DNA technology. Or maybe you can decide to say, okay, maybe you go for the animal studies. If the enzyme is present in the animals, you can slaughter the animal. You get the organ, let's say the enzyme is in the liver, so you get the liver organ from the animal. You open up you homogenize the liver, then you open up, you get the cells of the liver, then you open up the cells of the liver. After you open up the cells of the liver, then you have actually a chunk of proteins and also non-protein component. So from there, you can actually go ahead with the precipitation. So mostly we use ammonium sulfate precipitation. So this precipitation is a technique that when you add ammonium sulfate, all the protein component will clump together and then the non-protein component will be uh, will now actually be separated from the protein that precipitated. And this basis of precipitation is based on solubility, and also pH. Then what are the phase partitioning? That is with, let's say, chromatographic techniques. So after you have your protein precipitation, you can decide to say, okay, maybe you'll go for ion exchange chromatography. Maybe you have an idea your protein is negative, then you can actually go ahead with cationic exchange chromatography. Cationic exchange chromatography is a chromatographic technique in which it actually has a positive charge where the negative charge proton will bind to it. Or you can decide to say, okay, we'll undergo for hydrophobic interaction chromatography, or maybe to affinity or gel filtration, or reverse pay. So each of them have their own principle. So like for ion exchange chromatography is based on charge distribution. Hydrophobic interaction chromatography is based on hydrophobicity of the protein. Then affinity is based on the ligand binding. That is, you'll design a ligand that can actually perfectly fit and bind with your protein of interest. And then for the gel filtration is based on size and shape. And then for the reverse phase, high performance lipid chromatography, that's HPLC, is also based on hydrophobicity and the size. So this is actually some of the protein purification techniques. So the best thing that you do after you have your protein of interest, so you need to go and from the literature check whether your protein have particular ligand that can bind to it. Then you bind the ligand and then you go for affinity chromatography. And if it is actually positive or negative charge, then you can decide to say, okay, we'll go for ion exchange chromatography. And if it is based on hydrophobicity, then you can now say, okay, you just go for a reverse phase HPCL, that is high performance liquid chromatography. And then, of course, the next one is immobilized metal affinity. It's also based on the metal binding affinities. So this is actually some of the techniques. So all these are the chromatographic techniques: ion exchange chromatography, hydrophobic affinity, gel filtration, reverse phase, immobilized. They are all chromatographic techniques that you can use to separate your protein. Then we have electrophoresis, but it's not dogmatic because sometimes if you can actually identify a ligand that can perfectly bind to a protein or enzymes of interest, then you can just use affinity chromatography. You don't need ion exchange chromatography. You don't need gel filtration. You don't need reverse HPCL. Then for the next one, we have electrophoresis. So electrophoresis is actually to separate your protein based on charge size and shape. And then we have isoelectric focusing, which is also another technique which actually separate protein based on size, shape, and density. Then we have we have centrifugation, which separate protein based on size, shape. And then we have ultrafiltration, which also separate proteins based on size, shape. We have differential denaturation, which is based on thermal stability. So we also have affinity chromatography, which is based on biological recognition, that is binding. So ladies and gentlemen, this is just a brief description of protein purification steps. So what are the application of enzymes in biotechnology? So enzymes are extensively used in biotechnological applications such as the production of biofuels. So they play a key role in genetics engineering where they are used for cutting and joining DNA fragments. So these are some of the applications of enzymes in biotechnology. We can use it we can use enzymes in, in many biotechnological applications, such as biofuels. You can use it also in genetics engineering, where they use for cutting and joining DNA fragment. That is in recombinant DNA technology. And the enzymes are also used to utilize, or are also utilized in the food industry for processing, like fermentation and food preservation. So clinical application of biochemistry, of enzymology. Enzymes are important in clinical diagnosis. You can use enzymes to diagnose disease. So where enzymes level can indicate certain diseases. When enzymes level is high or low, it's an indication of a particular disease. So enzyme replacement therapies, you can actually produce this enzyme or you can isolate them, especially in some diseases that are actually occur as a result of deficiency of that enzyme. So you can use that enzymes to cure the disease. So that is enzyme replacement therapies, like Gaucher's disease. So understanding enzymes function can aid in drug development and and the design of enzymes inhibitors. So what are enzymes inhibitors, ladies and gentlemen? So enzymes inhibitors are substances that decrease enzyme activity, which can be reversible or irreversible. So we have reversible enzymes inhibitors and we have irreversible. So when we say reversible, it means that when you remove the inhibitor, the enzymes will still actually work. But in the case of irreversible, the enzyme's activity will not come back again. It will already destroy it, so it will not this thing. So what are the, we have different types of inhibitors. We have competitive inhibitors where the inhibitors bind to the active site. So the inhibitors and the substrate are competing with the same active site. That is why it's called competitive. While non-competitive inhibitors bind elsewhere on the enzymes, not on the active site. And we have these inhibitors are often used in drug design to target specific enzyme associated with disease process. So most of the actually uh, drugs that we have in the pharmaceutical industry, they are enzymes inhibitors. You study the enzymes, you study how the enzymes or how the protein is related to a particular disease pathogenesis. Then you design a drugs, you develop, you identify inhibitors that can inhibit that particular enzyme. And from there, you have actually started with the drug development. So this is how the enzymes inhibitors divide. So we have enzymes, we have reversible and irreversible. So under reversible, we have active, we have competitive, we have non-competitive, and we have uncompetitive. And under irreversible, we have active site directed and we have suicide inhibitors. So futures, future direction in enzymology. So advances in structural biology and computational modeling are enhancing our understanding of enzymes mechanisms. Research into enzymes engineering aims to create more efficient and specific enzymes for industrial application. The exploration of enzymes in synthetic biology could lead to innovative solutions for environmental and health challenges. So, ladies and gentlemen, this actually brings us to the end of this our lecture for today. And I hope you find this video very interesting. And if this is the first time you are coming to my YouTube channel, please subscribe and join the YouTube community.